Welcome, future doctors. So, if you're a first-year veterinary student staring down a massive histology syllabus right now, take a deep breath. You are exactly where you need to be. Think of this explainer as your ultimate visual atlas and high-yield study guide. We're going to cut through the noise and focus specifically on the body's wireless communication network, the endocrine system. We're keeping it strictly to the essentials you actually need to know to ace your exams, making these microscopic structures super easy to understand and, more importantly, remember. Let's get into it.
Here's our roadmap for today. We'll start with the general features of the endocrine system, look at hormonal control mechanisms, and then dive deep into the pituitary gland, its origin, anatomy, the anterior lobe, and the posterior lobe. Finally, we'll wrap up with a quick intro to the thyroid gland. All right, Section 1, General Features of the Endocrine System. At its core, the whole point of the endocrine system is maintaining homeostasis. That delicate internal balance an animal absolutely needs to survive. To really get this, let's compare it to the nervous system. Think of the nervous system like a hardwired, high-speed internet connection. It uses neurotransmitters over incredibly short distances for lightning-fast reactions. The endocrine system, well, it's more like a Wi-Fi broadcast. It sends out chemical signals, hormones, over long distances. The result is a much slower response, sure, but it's prolonged and way more widespread.
Moving right along to Section 2, Hormonal Control Mechanisms.
So how exactly do these wireless signals travel from point A to point B? We're looking at three main mechanisms here. First up is classic endocrine control. This is where hormones are secreted directly into the bloodstream to reach distant target cells. Then we have paracrine control. This is when a cell releases hormones that act locally, just on the adjacent cells right next door. And finally, autocrine control. This one is pretty wild. The cell secretes a hormone that actually acts on itself. It's literally talking to itself.
Now, this brings us to a massive, high-yield exam point, the blood supply. Remember, endocrine glands are ductless glands. Because they literally don't have ducts to transport their secretions, their cells arrange themselves in cords or clusters to maximize direct contact with blood vessels. An endocrine gland's efficiency relies entirely on being intimately glued to fenestrated capillaries. Fenestrated just means they have little windows or pores, allowing these large hormone molecules to easily jump straight into the bloodstream. Without this rich blood supply, the whole system just doesn't work.
Okay, Section 3, the pituitary gland, origin and anatomy.
Meet the hypophyses cerebri, better known as the pituitary gland. It's often called the master gland because it bosses around most of the other glands. Anatomically, it sits in a super protected, cozy little bony vault at the base of the brain called the sella turcica. But here's the irony. Even though it's the master gland, it actually takes its orders from the hypothalamus, which physically connects to it via the pituitary stalk. Now pay attention, because this right here is a classic test question, the dual embryonic origin of the pituitary gland. It literally comes from two completely different places. The anterior lobe, or adenohypophysis, actually originates from the roof of the developing mouth, specifically a structure called Rothke's pouch, and then pinches off. Meanwhile, the posterior lobe, the neurohypophysis, grows downward from the neural tissue of the brain and stays connected. Two completely different origins mean two completely different functions. Which perfectly sets up Section 4, the anterior lobe and its secretory cells. Let's zoom in on the histology of the pars distalis and the anterior lobe. When you look under the microscope, the cells categorize themselves based on how well they take up stain. First, you've got the chromophobes. Phobe means they fear the stain. They look pale. They make up about 50% of the cells, and they're basically inactive reserve cells. Then you have the chromophils. These guys love the stain. They make up the other 50%, and they are your highly active hormone-pumping factories. So, how do we tell these active chromophils apart? They split into two camps. First, the acidophils, or alpha cells. These stain pink and are PAS negative. Second, you've got the basophils, or beta cells. There are fewer of these, but they're larger and they stain blue or purple. But here is the golden nugget for your exam. Basophils are PAS positive. Why? Because the hormones they secrete are glycoproteins. Definitely memorize that. Let's break down what those pink acidophils actually produce. There are two main types. You have somatotropes, which secrete growth hormone, or GH, targeting bones and cartilage to stimulate body growth. And then you have mammotropes, which secrete prolactin. Prolactin targets the mammary glands to stimulate milk production. And what about the blue basophils? They have three main types, and they're all about controlling other glands. Thyrotropes secrete TSH to stimulate the thyroid. Gonadotropes secrete FSH and LH, which target the ovaries in females and testes in males to regulate reproduction. And finally, corticotropes secrete ACTH, which tells the adrenal cortex to pump out cortisol. Moving on to section 5, the posterior lobe, also known as the neurohypophysis. I need you to highlight, underline, and star this next point in your brain. The neurohypophysis is not an endocrine gland. It is strictly a storage site. I guarantee this will be a trick question on your histology exams. The posterior lobe produces absolutely nothing on its own. It simply stores neurosecretions that were actually manufactured way up top in the neurons of the hypothalamus. So when you're looking at the posterior lobe, or pars nervosa, under the scope, what are you actually looking for? 3 things. First, thousands of unmyelinated nerve fibers dropping down from the brain. Second, hearing bodies. These are these swollen, dilated nerve terminals packed with stored oxytocin and ADH. And third, pituicytes. These are basically supportive glial cells that provide the physical scaffolding for this whole storage operation. And finally, section 6, a quick introduction to the thyroid gland. The thyroid gland sits right there in the neck. Anatomically, it usually consists of 2 lobes connected by a little bridge called the isthmus. Histologically speaking, it's wrapped up in a double capsule that sends fibrous septa inward, dividing the gland into lobules. And crucially, those septa carry all those highly permeable fenestrated capillaries we talked about earlier right into the heart of the gland. We'll dive deep into thyroid histology in our next review, but I want to leave you with a question to chew on. We know these ductless glands completely rely on rapid diffusion into the blood. So, when the thyroid gets a massive signal from the pituitary and suddenly needs to pump out a ton of hormone, how do you think the epithelial cells lining the thyroid follicles actually rearrange their physical shape from a resting state to maximum overdrive to handle the load? Think about that incredible cellular shape shifting, and we'll unpack the answer next time.